Hi, welcome to Microscopy. Today we'll be talking about parts of the microscope as well as its operation. In front of you, you can see the Olympus CX33, a compound microscope. Listed are common microscope parts that can be seen on a variety of different makes and models. It is important to know the technical names of the parts as they will be used throughout this video and future labs. The most important of them are the ocular lenses, the stage, the light brightness control dial, coaxial stage controls, the fine focus adjustment knob, the coarse focus adjustment knob, and the arm. Microscope operation. There are several rules that should be followed to prevent permanent damage to the microscopes and ensure accurate results. 1. When transporting the microscope, it is important to hold it with two hands, one on the arm and the other on the base. These microscopes are heavy, delicate, and expensive, and any contact that is less than gentle can render them inoperable. 2. If you must clean any of the optics, use specified lens paper. Any other products can permanently scratch the glass, which ultimately distorts the view. 3. When you put the microscope away, and before you turn it on, make sure the objective lens is set to the lowest setting, in our case 4x, and the stage is raised to the utmost position. This will help focus on the slide and prevent damage to the objective lenses. 4. When using the higher magnification objective lenses, only use the fine focus knob. Because the distance between the objective lens and the slide become closer at higher magnifications, you can accidentally break your slide or damage the objective lens. It is also important to follow the given protocol for operating and viewing slides under the microscope. As long as the safety protocols have been followed, go ahead and turn on your microscope. This will turn on the light. You can adjust the light through the light brightness control dial. Left to darker, right to brighter. If your slide is on the stage, use the coaxial stage control to center the slide under the optics. You should then adjust the interpupillary distance of the oculars, also known as the eyepieces, as well as the diopter. These two adjustments can help resolve issues of stereoscopic viewing and clarity of your slide. To bring a subject under a microscope into focus, always start with the lowest objective lens and lower the stage using the coarse adjustment knob to bring the subject into view. Use the fine focus knob to sharpen the image. To increase the magnification, carefully grip the objective lens ring, rotating to the next highest objective lens, in our case, 10 times magnification. If the image was properly focused, only fine focus adjustment should be needed to bring the subject into clear view at higher magnifications. The maximum objective magnification we will use in this course is 40x, and only fine adjustments should be used to prevent damage. When you're finished with the microscope, return the objective lens to the lowest setting, in this case 4x. Carefully remove the slide by releasing the slide clip off of the stage. Make sure that the stage is raised to the utmost position using the coarse focus, and turn off the light using the on-off switch. Unplug the microscope, grabbing the base of the plug from the outlet. Some of you may be using the Olympus CX31 microscope as opposed to the CX33. Operationally, these work the same, although some of the parts are in different locations. The rules for handling the microscopes are the exact same, and care should be taken when handling or using the microscopes. The Olympus SC61 is a dissecting microscope and is different than a compound microscope that you have seen. While the safety and operation procedures are the same as the other microscopes, this microscope has one objective lens and two light sources, one emanating from the bottom with the control dial on the right, and one coming from the top with the control dial on the left. This will help to view large specimens that could not otherwise be seen under the compound microscope. There are two controls to help view your specimen. One is the magnification dial. This will increase the objective magnification by the numbers listed on the dial itself. The other is the focus dial. Unlike compound microscopes, there is only one adjustment for focus, and this knob will raise or lower the optics and top light. When viewing subjects under the microscopes, there are two concepts that are important to remember. First is that the total magnification is calculated by multiplying the objective lens magnification by the ocular lens magnification. All the microscopes in the lab have an ocular lens magnification of 10x. The second is that both the working distance and your field of view decrease with the increase in magnification. Again, only use the fine focus knob at higher magnifications. Once the specimen is properly focused and in view, analysis can begin. Now we will discuss cell division, with particular interest in the stages of mitosis within plant cells. There are 5 stages of the cell cycle. The longest and non-dividing stage is interphase, followed by the 4 stages of mitotic division: prophase, metaphase, anaphase, and telophase, as well as cytokinesis. All these phases can be seen within the slide of the onion root tip at 400 times magnification. Interphase can be identified by the nucleus clearly visible at the center of the cell. Prophase is identified by the condensation of the double-stranded chromosomes and dissolution of the nuclear membrane. This is sometimes referred to as a bowl of spaghetti. Metaphase is marked by the double-stranded chromosomes lining up on the equator. Anaphase can be identified by the movement of sister chromatids towards opposite poles of the cell. Telophase is identified by the chromosomes uncurling and the nuclear envelope forming. In plants, cytokinesis is marked by the forming of the cell wall.